For the Journal Club today, we're going to continue on with delving into the ancient history of immunology. And really what we're going to talk about today is the beginning of allergy as a concept, that you could have a bad reaction as a consequence of the immune system. And since it was the beginning of allergy, it was the end really of immunotherapy or serotherapy, essentially. And as you recall, Pasteur told everybody at the end in the 1880s that they were going to be able to prevent all the major contagious diseases by vaccinating and also treating infectious diseases even after the infection occurred, like his rabies vaccine. And so he was going to cure everything, but he dropped out of the scene around 1885. And then in 1895, he died. So between Pasteur in 1884, 85, and then 1890, then we had on the onset of von Behring and Kitasato, and they introduced the idea that Pasteur, I forgot to say, he thought vaccination worked because, and you needed a live organism, a live vaccine, so that it would deplete the host of vital trace nutrients, and therefore the host would not be a suitable place for the microbe to gain a foothold and replicate and live and so forth. And of course, in 1890, with von Behring and Kitasato, it became, for the first time, the concept was introduced that immunity was not a passive phenomenon that happened to the body, but rather immunity was something that occurred as a consequence of the reaction of the body with the introduction of foreign materials into the organism. So that was a big deal, actually. And from there on, after 1890 on, whatever the stuff was in the blood, I think the saying went that the blood was a very interesting juice. Whatever it was in there was a reaction on the part of the host, and it was somehow conferring immunity. So as a consequence of that, and because of the von Behring and Kitasato working on diphtheria and then tetanus. Diphtheria was a major problem, particularly in the pediatric group of people. And you couldn't be sure if you had a child that child was going to survive really past infancy and certainly past early childhood. Most of them died. If you ever get, maybe I said this before, but if you ever get a chance in New England, if you go up, just stop your car and go into a cemetery by the side of the road, and you can see the gravestones of whole families. So there'd be a mother and a father. Sometimes there'd be more than one wife because the wives died in childbirth. But then there was a whole bunch of kids, and most of them died before. You only about 10, 20% of them made it to adulthood, and the rest of them succumbed to these contagious diseases, and diphtheria was a major problem. And so after von Behring and Kitasato explained this reaction that occurred, and there was some antibody activity in the sera and the fluid portion of the blood, everybody started injecting things into people, into animals, and so forth, and trying to figure out exactly what was going on. And so the next paper that is on the website, it's from 1902, and it's Paul Portier and Charles Richet, who discovered really the first description of allergy. And the story is it's all in French on the website. And so if you can read French, then it's only through a few pages. And the story, though, is quite interesting because they were on a yacht out of Monaco with the Prince of Monaco, and the Prince said, "Why don't you guys study this venom that's coming out of the Portuguese man of war?" And so they did, and they started doing experiments on dogs, and they were injected the venom that they got from the tentacles of the Portuguese men of war, the jellyfish, and they found that if they injected, well, there was a dose-dependent toxicity of these toxins coming out of the jellyfish, but you could start lower and you wouldn't kill they were working in dogs. You wouldn't kill the dog with your dose if you used low dose. But then if you repeated that dose, particularly after a couple of 3 or 4 weeks, some of the dogs just absolutely had an acute reaction within minutes of the administration of the venom in the same dose, or even a smaller dose would cause this whole thing. They went through lots of dogs in their experiments, and they described what we know today as anaphylaxis. And I never understood the derivation of that term, and I just recently looked it up because I was going to give this journal club. Phylaxis, it goes back to is a Greek term that means protection. So anaphylaxis is, against protection, actually toxicity, and prophylaxis, if you remember the prophylactics that you used to use when you were a young blade, those were preventatives on the thing. So phylaxis is the stem word. Anaphylaxis is what happens is after you get that second dose of this, or after they gave the dogs the second dose of the venom, they went into essentially the blood pressure fell, and they also had an acute reaction in their respiratory tract, so that they became hyperventilating and short of breath and so forth, and they developed hives all over their bodies and so forth. So that's what anaphylaxis is, and that's what all of the drug companies now are worried about with coronavirus vaccines. And there have been reports now that that some people, I think the incidence is very, very low. It's like 1 out of 1 million or 2 out of 1 million. But one was written up in the press as a physician who had a food allergy to crustacea, the lobster, shrimp, and crabs. And I have that same allergy. And according to the New York State Health Department, that allergy, that food allergy, causes the largest number of cases of anaphylaxis, where you can die within minutes after ingesting lobster, crab, or shrimp. So you're at the cocktail party, and all of a sudden you take a bite of this, and and this has happened to me, and I know immediately that I'm in deep trouble. But I don't get anaphylaxis, but I do get deathly ill. So that was Pirquet and Schick in 1902. And Pirquet went on to get the Nobel Prize for the discovery and the description of anaphylaxis. I think, I can't remember, 1913. So the very next year, in 1903, there was another paper that came out by another Frenchman, and it's also on their website. And also, it's only 3 or 4 pages, and you can read that there too. It's Maurice Arthus, and he decided to see so they knew venom would cause this, and toxins would do this, but they didn't know that anything else might cause anaphylaxis. And so he embarked on a series of experiments using rabbits and injecting them with goat sera. And he found a very similar phenomenon. If he injected goat sera and then waited a couple of 3 weeks and then injected another dose, he could get anaphylaxis out of this whole thing. And therefore, other people went on to show that it wasn't just these toxic venoms from the Portuguese men of war from the Côte d'Azur down in France. It was any old protein that you might inject into these. If you did a repetitive injection, you risk developing an allergy, which would manifest itself by anaphylactic shock. Later on, that was basically shown to be due to an antigen-antibody reaction. Jeff Ravitch is an investigator at the Rockefeller University here, a molecular geneticist, immunologist, and he cloned the genes for the FC receptor of antibodies that allow phagocytic cells to recognize antigen-antibody complexes and engulf them and so forth, activates them as well. And that's the source of the what they call the vasoactive products of mast cells as well as macrophages that cause anaphylactic shock. So that was not good. It meant that Pasteur's dream or even it suggested that von Behring and Kitasato's dream of using serotherapy for diphtheria, for example, but people started doing it anyway. And then, as I think I said before, horse farms were developed, outside of Berlin for the Koch people, and then outside of Paris for the Institut Pasteur, where they would immunize these animals, these horses, because then they could bleed them. They could get huge amounts of sera that they could use in serotherapy. And that brings us to the report in 1905 by Clemens von Pirquet and Bella Schick. So Pirquet was an Austrian, and Schick was a Hungarian, but they both went to the same medical school in Austria around the same time, around the turn of the 20th century. So in 1905, they started a whole series of clinical, as soon as they got out of medical school, and they were on the same staff of the hospital in Vienna, they started treating kids with diphtheria with horse serum. And so this paper from 1905 is very interesting because, this goes back to what we discussed before, is that the English and the Americans were still killing Indians about this time. The French and the British were much further ahead in terms of their science, or the French and the Germans. And so this paper from 1905, which really this became the Bible of allergy. And it wasn't translated into English until 1951. And it was translated into English by Bella Schick, they did many experiments in Austria and so forth. He ultimately got offers to come to the United States, and he went to Mount Sinai in New York, and he was a Hungarian Jew. And he carried on many, many experiments and became, a celebrated professor in the United States. Meanwhile, von Pirquet stayed for the most part, he stayed in Austria, and he committed suicide in 1929 together with his wife. Now, I haven't delved into the history enough to find out what happened, but I'm just wondering it's because of the worldwide stock market crash and depression that happened in 1929. I mean, why else would you kill yourself? Maybe along with your wife. Anyway, Bella Schick was approached by the Williams and Wilkins Company, which publishes the Journal of Immunology in the 1950s, and they said, "Why don't you translate this paper? It's your paper." And so forth into English, so that the English-speaking doctors, they're having trouble with this paper, really getting the nitty-gritty from it and so forth. So he did it. Then they published it then in 1951, and that's what's on the website. And if you access that, you'll find out that it's 121 pages. So this goes back to Jenner in 1798, and then can't remember, the physicians having many case reports, and this is what the majority of this paper is all about. And I'm going to go into a little bit of this. It's the name of the paper is called Serum Sickness, or in German it's Serum Krankheit. In the preface of this paper that Bella Schick translated, he basically lays out that von Pirquet was really a dear friend, and they had, and these early experiments they did, they were just young blades back in the turn of the 20th century. And in the introduction to this paper, he goes through a little bit of the history of the physicians trying to treat diseases with blood and blood products and so forth. And he points out that transfusions were made, between people and also in experimental animals in the years and centuries before, and this is before you could do transfusions in human beings, and it was finally figured out after the 40s that transfusion reactions, which I'll describe in a second, only happened when you had a mismatch between the donor and the recipient. It had to do with red blood cell surface molecules that would be destroyed by sera from another individual. So they tried blood transfusions, but that was to say, what would happen then would be is that you put blood into another animal, and all of a sudden there'd be this acute onset of hemolysis, so that the red blood cells were essentially destroyed, and the hemoglobin was which causes the cell to be red, would be released into the bloodstream and go into the kidney and then into the urine, and you get hemoglobinemia and hemoglobinuria, and essentially you get hugely high fevers up to 105, 106. You get disseminated intravascular clotting, and basically you die. And it's not a good way to go, essentially. So that was using blood to transfer from one individual to another. After the von Bergen-Kitasato, they got rid of the red blood cells and eliminating that problem. And they found that they could really transfer serum from one individual to another, and it was okay. And you wouldn't get any bad reactions. And the thing is, and this is what they first started using it for diphtheria in the 1890s and using the horse serum. And the thing is, as I've said before, is it worked. Kids that were really destined to die because they were gasping for breath and so forth, when they got this horse serum, they got better really quickly, within 24, 48 hours, and that was good. Only problem is that, and that's why the name of this paper is called Serum Sickness. Later on, they had a reaction against the horse proteins, the proteins in the horse serum. But after 1900, they went on to treat people in the business, went on to treat other kinds of infectious diseases, scarlet fever and so forth. And they used large doses of serum. I mean, 100 or 200 cc's or milliliters, and that's a lot. A glass of wine is a couple hundred cc's. So that gives you an idea that they were sticking in. And the thing is the Pirquet and Schick, they were treating little babies and small children, and they were intraperitoneally, they were injecting huge amounts of horse serum into these little kids, saved their lives, which was really good. So they decided to write up they did a series of experiments about what happened after you gave the horse serum to these kids. And chapter 1, they summarize the clinical aspects of what they called serum sickness. And they have this, they gave examples in case reports, very detailed case reports as to what happened to all these little kids. And I won't, I'd spend a long time doing it, but the general clinical picture was that there'd be no symptoms for like 8 to 10 days. The kid was fine, got over the diphtheria or the scarlet fever and so forth. But then there would be, after about 10 days, there'd be an onset of hives. Basically would go all over the body and be tremendously itchy. You have a central blanched out central zone and the surrounding erythema, redness, and itches like mad. You'd get a high fever. You get a tachycardia. The heart would start racing. You get local lymphadenopathy. Wherever they placed the serum. And then it would be disseminated, and you get enlarged lymph nodes all over the body. And then the patients would develop edema. Their tissues would fill up with fluid, and that was because the kidneys were basically affected by this whole deal. And what they figured out later on is that the kidneys were being clogged up by antigen-antibody complexes. So they had abnormal kidney tests, function tests. They could look at the urine and they could find that there was a lot of protein in the urine and they shouldn't have been in there. You get disseminated arthritis, and all the joints of the body started really aching and so forth. And that was subsequently shown to be due to antigen-antibody complexes. And that happened to me once, actually, when I was a young doctor. I got German measles, rubella, and I was sick. Oh, this was right before they came out with the vaccine for rubella. And I developed this arthritis, arthralgia all over my body. I thought I was going to die. And then after a while, I didn't care if I did. In fact, I was sort of thought it'd be a good idea, you know? So, so they described that, and that was the serum sickness. And those are the clinical manifestations of this, since these guys were doctors. And then they have, so that was chapter 1. Chapter 2, they re-injected these patients with the same sera that they had done the first time around. And this time they got an accelerated reaction. And you didn't have to wait for 8 to 10 days. You had an immediate general reaction, which was anaphylactic shock, just like had been described by the previous guys. And then there was a little bit of a delayed kind of thing, but it wasn't as delayed as 8 to 10 days. It would happen 2 or 3 days later. You'd get and the serum sickness clinical picture would just be heightened. So then they did a bunch of studies, and they summarized in chapter 2 or chapter 3 the theory that they'd come up with to explain this whole thing. And so the first is they said, well, it's obvious because of the first and second reaction and how the differences in the timing of those things, that the host was altered by injecting these proteins, these horse proteins into them. And they developed an in vitro assay for them that they could put a microscope slide and they could mix the sera from the patient with the serum from the horse, and they could see that there were flocculants. It was agglutinated. They were, as you could, under the microscope, you didn't necessarily couldn't see it with macroscopically, just with the naked eye, but under the microscope they could see these little precipitates forming. So they called these things precipitins. And that was the beginning then of looking at what the heck was these things in the serum. And they had different kinds of tests. They had some tests where they could show that the serum of a patient or an experimental animal that hadn't been injected with a particular antigen. They went to, they started with red blood cells. For example, they would put sheep red blood cells into dogs and so forth. And then the recipient would develop what they called hemolysins. So that these were, now we know, antibodies that actually lysed the red blood cells. And another word that was coined in this instance were hemagglutinins that would agglutinate the red blood cells, and so that you could see that under the microscope. So, and there were several more assays that were developed on the function of the serum from these in these experiments. And it's exactly the same thing that happened almost 100 years later when we started trying to understand what was going on with the cytokines. So interleukin-2 and interleukin-1 and all those things. Initially, they were discovered because we discovered that interleukin-1 was a lymphocyte activating factor, and they developed an assay for that function. And then we developed the assay for T cell growth, and that was T cell growth factor. And then it went on and on. We had B cell activating factors and all kinds of things happened. And so that the activity then was the way that the early serologists and the early immunologists could follow their antibodies, and they developed quantitative assays so that they could find out the concentration of the antibodies in the serum. And this was all very good. And the other thing they said in terms of the theory of their serum sickness is that after a while, the antibodies that they could detect with their assays seemed to disappear from the serum. Yet that person or that animal was still immune, because if you re-injected them, what they found is that the antibodies would reappear much more rapidly second time than the first time. And that essentially became the first descriptions of immunologic memory or the so-called anamnestic immune response, the secondary immune response occurring much more rapidly and going to a greater height than the primary immune response. And that's why we're in the COVID times. That's why we have the initial dose and then the booster dose a month later, because after the first reaction, you get a certain amount of what happens is that the lymphocytes, both the B cells and the T cells that have receptors for this particular antigen, let's say it's COVID virus, there aren't very many, you know, like 1 out of 1 million in the body initially, but then after you have, then they proliferate, so they expand massively in number. And then so that the second time around, you simply got many more lymphocytes that were programmed to have their antigen receptors react with that particular antigen. And that's, so in this paper from 1905 on Pirquet and Schick, essentially came to the conclusion, and in their interpretations of these phenomena, that there must be other types of immune reactivity that had developed in the body that didn't have anything to do with antibodies. And of course, that's what we know as cellular immunology today. In a very short period of time, only 15 years in the beginning of at the turn of the 20th century, this paper really lays out a lot of the fundamental characteristics of immunity and the immune response and immunology. It then took another 100 years, bring us up to date now, to try to figure this all out and to find out what was going on. And that's one of the reasons that I think that it's important to read these early papers, because the timing of the discipline of the science that you might be studying is really important, and knowing who the major contributors were and learning a little bit about their lives. That's why you can go on Google now and look, just put the person's name in, and you can get all kinds of biographical information about them, which I find very interesting, because you know, as I've said before, science is done by human beings. It's not just something you read in a textbook in a short paragraph that summarizes 100 years of experiments. They end up saying, now remember, this is 1905. They said, because of the fact that you could get this kind of an immune reaction to serum from the horse into the human, whatever, vaccination as a phenomenon doesn't depend on using a live organism that will deplete the host of these trace nutrients, as said by Pasteur. So they knew this in 1905, that you can get an immune reaction to all kinds of things, and not even microbes, just proteins from another animal or another person. The other thing that they pointed out is that you didn't have to have a replicating antigen. You didn't have to have a replicating microbe to generate this kind of reaction. You could just put the static molecules into these animals and get the same thing. So that was the setup then for the beginning of the end of immunotherapy as a clinical discipline, and the beginning of immunology as a basic science. And the immunology as a basic science, really, for the first 50 years of the last century, between 1900 and 1950, the name of the game was to find out what these antibody molecules were that were causing all these phenomenon that they could find from doing these kinds of clinical experiments or animal experiments. That essentially then, the whole first half of the century was taken over by chemists. So a whole new discipline of immunochemistry came about. And the immunochemists then, they were at the top of the food chain. They looked down their noses at anybody that wasn't a real chemist that was trying to find out the nature of antibodies and how did the functional aspects of things that people had found, how that was mediated by these different antibodies. And of course, nowadays we know that antibodies are molecules that are called immunoglobulins, and there's 5 different classes of them and so forth. And we'll get into that as time goes on in the journal club. And so I think I'll stop there. Let me see if yeah, that's it. I don't have anything else. Thank you. So if you've enjoyed this video, Please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.